So pH and its importance.
Mm.
Uh, a full form of pH is potential of hydrogen. It is also known as the negative logarithm of hydrogen ion concentration. It determines the strength or power of hydrogen. The hydrogen ion concentration vary from less than 10 to 14 millimoles per liter to more than 1 moles per liter. Increase in one pH point results in 10-fold decrease in hydrogen ion concentration. So therefore, pH merely is a reflection of hydrogen ion concentration. What is an acid? Compound which forms hydrogen ions in solution are called acids. So they are basically proton donors. Bases are compounds which combines with H plus ions. So they are proton acceptors. Strong acids are compounds which gets completely ionized in solution, whereas weak acids and bases are compounds which are partially ionized in solution. A buffer is a compound that limits the change in hydrogen ion concentration when hydrogen ions are either added or removed from the solution. Coming to the importance of hydrogen ions, the hydrogen ions are highly reactive. They'll combine with bases or negatively charged ions at very low concentration. The proteins in our body they contain negative charged ions. Any change in pH will alter the ionization of the protein. The enzymes in our system are basically protein, so it will alter the degree of ionization of the enzymes and it will alter the functionality of the enzymes. So basically the enzymes function optimally around a physiological pH. If a graph is drawn against the enzyme activity and the pH, it is usually a bell-shaped curve. Coming to the hydrogen ions, the production of hydrogen ions is from the process of metabolisms, various metabolisms such as oxidation of amino acid, anaerobic metabolism of glucose to lactic acid and pyruvic acid. The majority of hydrogen ions is produced from CO2 release from oxidative or aerobic metabolism. Uh, uh, that is, CO2 plus H2O gives H2CO3 bicarbonic acid, uh, 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 which then dissociates into bicarbonate plus H plus. So this is how H plus is produced. Then coming to the control of H plus concentration in the body is by either CO2 elimination and renal handling. Now we'll see both in detail. Coming to the CO2 elimination, here there is a role of red blood cells. Basically, at tissues, the deoxygenated hemoglobin binds with CO2 and H plus. Um, at the tissues, the CO2 produced moves into the RBC via a concentration gradient. In the RBC, the CO2 plus H2O gets converted to H2CO3, which then dissociates into H plus plus bicarbonate. The H plus combines with reduced hemoglobin, and bicarbonate is exchanged with chloride. So as CO2 is responsible for majority of H plus ion produced by metabolism, the respiratory system is the single most important organ system in control of hydrogen ion. Here PaCO2 is inversely proportional to the alveolar ventilation. So this happens in the respiratory system. Next, coming to the renal handling of bicarbonate and H plus. In the renal system, there is regeneration of bicarbonate, there is excretion of H plus, and also role of electrolytes such as sodium and chloride also play a role uh, in maintaining the bicarbonate and H plus. Bicarbonate is freely filtered in the glomerular uh, convoluted tubule. All the filtered bicarbonate is reabsorbed by all healthy individuals. In excretion of H plus, H plus is actively secreted in proximal convoluted tubule and distal convoluted tubule. Actively means this process is an active process which requires energy in the form of ATP. by the role of electrolytes, the sodium by the action of aldosterone, there is sodium reabsorption, and which is reabsorbed in exchange of H plus and potassium ions. The chloride is exchanged with bicarbonate. As these are the two most abundant negative charge ions in the plasma. Coming to disorders of H plus ions and homeostasis, first is respiratory acidosis. Respiratory acidosis is PaCO2 more than 6 kilopascals or 45 mmHg. Causes can be either due to inadequate CO2 excretion or excess of CO2 production. In inadequate CO2 excretion is due to decreased alveolar ventilation. So it can be either central causes, peripheral causes, or any lung disease which is causing inadequate CO2 excretion, and also any chest wall causes. Excess CO2 production is a very rare condition which is sometimes seen in malignant hyperpyrexia. Next, coming to respiratory alkalosis, it is seen in hyperventilation due to anxiety. Next, coming to metabolic acidosis. Metabolic acidosis can either be due to excess of H plus production or excess of bicarbonate excretion. In excess of H plus production, the main thing here is due to tissue hypoxia, which results from anaerobic metabolism. So the tissue hypoxia can occur due to different causes. It can either be decreased arterial oxygen content as seen in severe anemia, then hypoperfusion to the tissues, which can occur in conditions like cardiogenic shock or hypovolemia, or local causes such as ischemic limb or ischemic bowel, or decreased ability to use oxygen, which is seen in severe sepsis or cyanide poisoning. Diabetic ketoacidosis also. There is basically lack of insulin, so the glucose is not utilized. The cells acquire energy from the fat. Uh, the fat metabolism results in the production of ketone bodies with H plus ions. So this results in metabolic acidosis. Um, uh, Excess H plus production can also be due to inadequate excretion of H plus by the renal tubular cells, which can occur in renal tubular dysfunction. And we all also saw that aldosterone has an indirect action on H plus excretion. So hypoaldosterone also can cause inadequate excretion of H plus, as seen in Addison's disease, and ingestion of acids, which is a very rare cause. Next, coming to excess bicarbonate excretion. The intestinal secretions are very rich in bicarbonate. So, diarrhea, as seen in cholera or Crohn's disease, can result in metabolic acidosis, and also use of acetazolamide, as it is a carbonic anhydrase inhibitor, and bicarbonate is not reabsorbed. In metabolic alkalosis, it can either be due to excess of H plus loss or excess reabsorption of bicarbonate. So excess of H plus loss can, since H plus is maximally seen in gastric secretion, so vomiting can result, or prolonged vomiting can result in excess H plus loss, which can result in metabolic alkalosis, as seen in pyloric stenosis or anorexia nervosa. Excess reabsorption of bicarbonate, if chloride loss is more, which is also seen in prolonged vomiting, can result in metabolic alkalosis, and also use of thiazide or loop diuretics can also result in metabolic alkalosis. The other cause is ingestion of alkalosis, which is a very rare thing. Then coming to the compensatory mechanism, there are two main compensation. One is respiratory compensation and renal compensation. Respiratory, the respiratory center in the brainstem, it responds rapidly to changes in the CSF pH. Any change in plasma pH or PaCO2 results in a change in ventilation within minutes. So it compensates rapidly. Whereas in renal compensation, the kidneys respond by altering the amount of bicarbonate reabsorbed and H plus excreted. This usually takes for for 1 or 2 days. Yes, sir.
Okay. So you have dealt more with the pathological conditions of pH. Okay? But this question is more from the physiology angle of pH. The question when they ask what is pH and what is its importance in normal life. What is the normal range value for pH?
7.35 to 7.45, sir.
Did you mention that?
No, sir.
No, number one. Who introduced the term pH? Anybody in the class can answer who introduced the term or this acronym pH? Kiranmayi, Saundarya, Sagar, Shobika. It is by a Danish chemist called Sorensen. Sorenson. Sorensen. So being a Danish individual, Dane, he actually said, even though the English meaning of what he said, like power of hydrogen, is correct, the actual original description was in Danish language, and that was written as puissance hydrogen. Puissance, Puissance. Puissance means the power in Danish language. Puissance hydrogen was the actual term that was coined by Sorensen in 1909. So he was a Danish chemist. He was not an anesthesiologist nor a doctor. He was a chemist who described this. And in chemistry, all of you know, number 7 is considered as alkali or acidic?
Neutral.
Neutral, excellent. And below 7 is
Acidic,
Acidic.
sir.
acidic. Above 7 is
Alkalotic.
alkali. So your normal pH is acidotic or alkalotic? 7.35 and 7.45 you said. Is it on the alkaline side or on the acidic side according
Alkaline
to
side.
chemistry?
Alkaline side, sir.
It is on the alkaline side. Why blood is, instead of being neutral at 7, why is it being at 7.35 and 7.45? What is the reason? That is the importance of pH. Okay, that is the answer for the importance of pH. Okay. Why our blood is slightly on the alkaline side and not on the exact neutral state of 7? Anybody? Okay. I'll share my screen and describe that to you. Why you want to do that? Let's see that. So how pH is measured and maintained. That is not I forgot to include that, or other whether I asked or not. It is defined as the negative logarithm of hydrogen ion concentration. P means power in English, but puissance in Danish language, hydrogen. So the hydrogen ion concentration is the actual pH. That's why we write the first P as small letter and H as capital letter. You should never write both capitals. pH should never be written as both P capital and H capital. It should be P small and H capital. So small changes in the pH can cause major disturbance. Most enzyme systems function only within the narrow range of 7.35 to 7.45, especially the catecholamines. That is why in acidosis, when you give your catecholamines, they are not effective. Only a catecholamine which you can give is NORAD. Other dopamine, dobutamine, all these things, they don't work in severe sepsis and acidosis because of this reason. Okay, enzymes lose their function. And acid-base balance can also affect electrolyte changes. It affects membrane function, alter protein function, and also affect hormones. Okay, so that is the main importance of that. And Another important reason why it is on the alkalotic side, slightly 7.35 to 7.45, is life is an acidogenic process. There is an enormous quantity of hydrogen ion produced by each and every biochemical change that takes place in the body. So if your body is maintaining, if your blood is going to maintain a pH of 7, it will easily drift back to the acidotic side very easily because of the enormous quantities of hydrogen that are produced. That is why nature has made it such a safe margin that your blood will be slightly alkaline to neutralize the hydrogen ions that are produced instead of keeping it at an exact neutral value of 7. Okay, that is another reason that you have to know. So if you see the older 6th edition Wiley, the chapter on acid-base balance starts with this particular sentence: life is an acidogenic process, which means hydrogen ions are produced in enormous quantities in the body. That is the reason why your blood is slightly alkaline and not at the neutral value. And it is measured by using this pH electrode, which sometimes may be asked as an individual question itself. So this is having a reference electrode and the measuring electrode. All of you have to know about it. Sometimes this is also asked in the IVA also. How does your pH calculation done? Nowadays we have pH meters available at the bedside. Earlier we have to take the article sample, send it to the lab, wait for the result. Nowadays we have bedside pH measurement equipments are available. So the regulation to maintain the pH between 7.35 to 7.45, there are three primary mechanisms. One is the buffer system, second is the respiratory regulation, third is the renal regulation. Okay, so these are the three important mechanisms to maintain the value between this 7.35 to 7.45. Now, what are the, coming to the first mechanism, they are the first line of defense against the pH change. And they react very rapidly within seconds. And the buffer systems are blood, tissue fluids, and cells, which immediately combine with acid or base to prevent the excessive changes in pH. But it do not eliminate the hydrogen ions. They only neutralize, add them to the body, but only keep them tied up until balance can be re-established. So what are the three major buffering systems are? One is bicarbonate buffer, two is phosphate buffer, third is the protein buffer. So buffer systems, you can again classify them as those present in the extracellular fluid and the intracellular fluid. In the extracellular, you have the bicarbonate and protein. Of the protein, you have hemoglobin, amino acid buffer, and plasma buffer. And in the intracellular fluid, you have the phosphate buffer system. So three main systems of buffering, and this is again a physiology question. This is asked repeatedly in the exam. And the bicarbonate buffer system, how does it do? It takes the hydrogen ion and converts it into carbonic acid, which is a weak and stable acid. And that can break down into water and carbon dioxide, and it can be eliminated through the lungs. So it can be in the form of sodium bicarbonate, which will form carbonic acid. It dissociates into hydrogen and bicarbonate if necessary, or it can break down into carbon dioxide and water depending upon the change that happens. And the importance of bicarbonate buffer is it is present in high concentration, almost 40 to 50% of the, that is why it is given as the first option for neutralizing or maintaining the pH. And we have an alkali reserve. The ratio of bicarbonate to carbonic acid is 20 to 20:1. So the weak carbonic acid is quite unstable and it will be broken down by the carbonic anhydrase immediately. So the ratio between carbonic acid and the bicarbonate is 20:1. The concentration of the component can be regulated by the base constituent bicarbonate, that is regulated by the kidney, or by the carbonic acid, which will be eliminated as carbon dioxide and respiratory regulation. So this is the advantage of bicarbonate. It can work through the kidney or it can work through the lungs. Both the things can be done by the bicarbonate. Hemoglobin buffer. Next is a protein which is a hemoglobin buffer. This also plays a very important role in the respiratory regulation of pH. We transport metabolically carbon dioxide from the cells to the lungs for excretion. As hemoglobin releases oxygen, it gains the affinity for hydrogen ion. So at the tissue level, the RBCs take up the carbon dioxide, which again mix with water to become carbonic acid. Carbonic acid is immediately broken down to hydrogen and bicarbonate. This hydrogen will combine with the hemoglobin. It will be carried as hemoglobin combined with hydrogen at the tissue level. Once it comes to the lung, this hydrogen ion and oxygen will be released. HBO will be, it takes up the oxygen, releases the hydrogen. Again, there bicarbonate will add to that. It will break down into carbon dioxide and water. So this is what we saw in the last class, few classes earlier as Haldane effect and Bohr effect. Okay, Haldane effect at the tissue level and Bohr effect at the lung level is another way of asking this question. And the phosphate buffer, it is also an intracellular buffer. There is a disodium hydrogen phosphate, which can take up the hydrogen ions and become sodium dihydrogen phosphate and release one sodium. So it is an important, it is less effective than the RBC, but it is occurring in the renal tubular fluid and the intracellular fluid. And this is the second best mechanism. And the third protein buffer mechanism, the blood plasma proteins, especially albumin also is a very important buffer system. Contains a lot of CCOH and NH2 groups, which also take up the hydrogen and convert it into what is called the imidazole groups of histidine. So these are the various buffering systems. Now the respiratory compensation is the second line of defense, because based on the speed with which they all act. So the buffer system is the earliest. The second best line of defense is the respiratory mechanism, which again we saw as by hydrogen ion being taken up with the bicarbonate, converting into carbonic acid, coming to the lungs and getting broken down to carbon dioxide and water and elimination. So patient with acidosis, what will be his respiratory rate?
Tachypnic, sir.
Tachypnic. That's a method of eliminating, hyperventilating, and washing out the carbon dioxide to bring back the pH to normal. Okay. So that is, you if you remember that, you can remember this particular mechanism of maintenance. So cell production of CO2 increases, CO2 combines with water, comes to acidosis, so pH drops. This stimulates the respiratory center in the medulla oblongata. Rate and depth of breathing increases, CO2 gets eliminated by the lung, pH goes back to normal. So this is the mechanism of respiratory control. Now the chloride shift that happens in the RBC is another mechanism where carbon dioxide and again form the carbonic acid, again broken down. So to retain the bicarbonate, chloride is being taken. What is this phenomena called? What is the other name for chloride shift?
Hamburg.
Hamburger phenomena. Very good. That is another important physiological mechanism. So these are all the respiratory mechanisms. Coming to the renal mechanism, the role of kidneys in maintenance is mainly to preserve the bicarbonate in the and our elimination by kidney. It tries to eliminate hydrogen ion in the urine. That is why our urine is always acidic. Okay. Whereas your blood is alkaline, your urine is acidic because body wants to push out hydrogen ions as much as possible and retain bicarbonate ion as much as possible to maintain this alkalinity of the blood. So if you understand that basic point, it will be easy to remember about the pH. So the role of kidneys is maintenance is highly significant, but it's a very last mechanism or a late mechanism, which will happen not immediately, but after 24 to 48 hours. tries to provide permanent solution. So this is the ultimate way it can settle your pH issues, and this is the best mechanism. Later on the latest hour, this is like Rajini. It will come late, it will act well. And this is in contrast to temporary buffering, and kidneys regulate blood pH by maintaining the alkali reserve. Right? So urine pH normally lower than blood pH because it is about 6, and clearly indicates that kidneys have contributed to acidification of urine when it is formed from the blood, which is having a pH of 7.4. So in other words, hydrogen ions generated in the body, the normal circumstances are mainly eliminated by acidification of the urine. And hence the pH of urine is normally acidic, while that of blood is alkaline. So urine pH, however, is variable and may range between 4.5 to 9.5 depending on the concentration of hydrogen ions. And the 4 main mechanisms how the kidney helps is, main mechanism is excretion of hydrogen, reabsorption of bicarbonate, and excretion of other titrable acids and ammonium ions. This is another important mechanism. So if you see in the proximal convoluted tubule, this is the proximal convoluted tubule cell where the carbon dioxide water combines to form carbonic acid, breaks down into bicarbonate and hydrogen ion. Bicarbonate is reabsorbed back into the plasma, and hydrogen ion is eliminated in the tubular lumen along the urine filtered fluid. And sodium is reabsorbed in exchange because both are positively charged ions. Electroneutrality has to be maintained all the time. So for giving up one plus positively charged hydrogen ion, one sodium ion will be reabsorbed. So sodium and bicarbonate are taken back in the proximal convoluted tubule itself. Once the fluid passes from the proximal convoluted tubule to the tubule, that is the descending loop and ascending loop of Henle, which are called the tubular cells. There what happens? Again, sodium is reabsorbed from the filtered fluid, where the sodium bicarbonate is filtered, and this sodium bicarbonate takes up the hydrogen from the cells, combines it with bicarbonate, to form carbonic acid, breaks down to carbon dioxide. Carbon dioxide can diffuse easily through any cell membrane. It will come back, and in the tubular cell, it joins with the water again. The same reaction happens, and then it breaks down to bicarbonate. Again, bicarbonate is taken back, and hydrogen is given back to the lumen. So this is the mechanism at the tubular level. So PCT level, tubular level. Then the third mechanism is again disodium hydrogen phosphate. Earlier we saw sodium bicarbonate, how it is working. Here it is the disodium hydrogen phosphate, which breaks down into sodium and sodium hydrogen phosphate, which takes up the hydrogen to become sodium dihydrogen phosphate. This is the second mechanism at the tubular level. Again, sodium and bicarbonate are taken back into the body. The third mechanism is ammonia excretion. So this is NH3 is ammonia, which takes up one hydrogen, becomes ammonium, MOAMMONIUM. So ammonia becomes ammonium by taking up hydrogen. So there are 3 mechanisms happening at the tubular level. One is the sodium bicarbonate level mechanism. Second is the disodium hydrogen phosphate mechanism. Third is the ammonia mechanism. So all these 3 takes place at the tubular level, and by these things they were able to eliminate the hydrogen and take back the bicarbonate and maintain the blood at the level of 7.35 to 7.45. Okay, and this is the question I asked in the beginning. At a pH of 7.4, ideal for biological processes, and most important in blood oxygenation. So your oxygen carrying is mainly because of the slight alkalinity, and that is why in pregnancy, maternal what is the PCO2 level in the mother? pregnant woman? Is it 40 or less than 40?
Is
Below.
They are
alclose,
slightly
less than
on
40.
the respiratory alkalotic side.
Alclose, sir. Yes, sir.
And that is why the maternal pH, where PCO2 is slightly low and their pH is always on the slightly alkalotic side.
The
Could be
double
the
bore
positive.
effect, sir.
To release the oxygen to the baby as well as to take up the carbon dioxide. Okay, so many intermediates of biochemical reactions are also ionized at neutral pH, so this will not work. So utilization of intermediates will be difficult if the pH is at 7. So the oxygenation and enzyme activity will be best only at the level of the
The fetus also stays in a relative hypoxic condition in order to take up more oxygen. And the mother hyperventilates. That's why they have the double bore effect, sir.
correct. So that is the. So what you need to write about this question on pH and its maintenance only this. You need not describe all the pathological conditions of alkalosis, acidosis, metabolic, respiratory. All these things are not required. Okay. So please
Okay.
look into the
Sir.
question properly, and if the question is worded, what is pH and what is its importance, you have to write only about how the pH is maintained at this level and not required to change to abnormal pathological states. Okay, that is not
Yes.
required.
Yes, sir.
Okay, Sanjutha?
Yes, sir.
Okay, right.